Hello everyone. Today we will study about sexual reproduction in flowering plants, which is chapter number 2 in 12th class NCERT biology book. In part 1 of the video we have discussed pre-fertilization events in flowering plants and their reproductive structures which are found in the flower. And in today's video we will discuss about the fertilization events as well as the post-fertilization events. Let's start it. Events in the plant reproduction. Events in the plant reproduction are classified majorly into three categories, that is pre-fertilization events, fertilization events and post-fertilization events. Pre-fertilization events include gametogenesis and pollination, which is the gamete transfer. In gametogenesis, gametes will be formed in the plant body. Male gametes will be formed in male reproductive organ and female gametes will be formed in female reproductive organ. And we have done this in the previous slides, that in the part 1 of the video, in which megasporogenesis and microsporogenesis and the process, or we can say the process of pollen grain formation and embryo sac formation, female gametophyte formation, have been discussed. Then the various ways by which pollen grain get transferred from the anther to the stigma of the flower were also discussed there. Now, fertilization events. Fertilization event include the fusion of male and female gamete, and this includes two processes, double fertilization and triple fusion. Whereas post-fertilization events involve zygote formation, embryogenesis and seed formation. Now, during fertilization, the gametes will fuse. But as after pollination, or we can say during pollination, the fertilization will take place after pollination. And during pollination, the pollen grains which lie on the stigma, they will firstly germinate. Here you can see in this slide, in this picture, in this diagram, pollen grains are germinating and a pollen tube is formed. This pollen tube carries two male gametes, and this pollen tube enters the ovule through micropylar region, and this is guided by filiform apparatus of the synergids to the egg cell. Now in this slide, pollen germination is explained. This picture depicts that how pollen tube is developed. Here you can see pollen grain, which is a binucleate structure having a generative nucleus and the tube nucleus. This generative nucleus will divide to form two male gametes or two sperm nuclei, and this tube nucleus will guide the growth of the pollen tube towards the ovary. These all events will take place if the pollen and pistil interaction are compatible. Means if the pistil or the stigma part of the female reproductive organ allow the pollen or have the compatibility for the pollen, then the pollen tube will form, then the pollen will germinate. But if the pollen and pistil are incompatible with each other, then the pistil reject the pollen, it prevents the growth of the pollen tube, it prevents fertilization, and therefore pollen-pistil interaction can be defined as a dynamic process of pollen recognition followed by promotion or inhibition of the pollen. The chemical components of the pollen interact with those of the pistil during these interactions. Now, artificial hybridization. This is a process in which, if we have the knowledge of pollen-pistil interaction, we can manipulate or we can cross different species of plants to produce superior varieties, and that process which is used for producing superior varieties is known as artificial hybridization. Now, the process in which desired pollen grains are used for pollination and fertilization. Here you can see in this diagram the process of artificial hybridization is shown. The steps are shown. One is emasculation and bagging. What are these steps? We will discuss in the next slides. Steps of artificial hybridization. First step is emasculation, which is removal of anthers from the bisexual flowers of the female parent plant. आपने इसमें से एक bisexual flower है आपके पास. Here you can see in this diagram anther and filament is shown to you, stamen part and stigma part. इसमें से बेटे stamen अगर आप remove कर देंगे तो that will be the emasculation. And this is usually done before the anther matures, and this technique prevents self-pollination. Now next step is bagging. Bagging involve covering of the emasculated plant. हमने जो emasculation कर दी उसके बाद फूल को हमने cover कर देना है ताकि contamination ना हो, ताकि कोई भी undesired pollen with the help of pollinating agencies like wind, birds, insects or animals इनके थ्रू आकर स्टिग्मा पर लाई ना करे, अनवांटेड पोलन से पोलिनेशन ना हो. सो वी कवर इट विद द बैग, और जब जो डिज़ायर्ड पोलन है, जो डिज़ायर्ड एंथर्स हैं, वो मैच्योर हो जाएंगे, आप वहाँ से पोलन ग्रेन्स लेंगे एंड डस्ट दीज पोलन ग्रेन्स ऑन द स्टिग्मा ऑफ द फ्लावर एंड देन रीबैग इट. दोबारा से इसे बैगिंग कर देंगे और टैगिंग कर देंगे बेटे. हियर यू कैन सी इन दिस डायग्राम टैगिंग इज ऑल्सो डन. Now what is fertilization? Fertilization involves fusion of male and female gametes, and in flowering plant, two male gametes which are formed in the pollen tube enter into the cytoplasm of synergids through micropylar end of the ovule, and inside the ovule following will happen. One is syngamy, which is the fusion of gametes. One male gamete will fuse with the female gamete, that is egg cell. A zygote is formed, and this zygote later on develops into embryo, and this fusion is known as syngamy. Now next, triple fusion. As I have told you that during fertilization two events will take place. One is syngamy or double fertilization. Event B मैं बताऊंगी आपको क्या होता है इसमें. And other one is triple fusion. Now what is triple fusion? एक मेल गमीट जो पोलन ट्यूब में बना था वो बेटे फ्यूज हो गया एग सेल के साथ. नाउ द सेकेंड मेल गमीट दैट विल फ्यूज विद द पोलर न्यूक्लियाई एंड इट विल रिजल्ट इन फॉर्मेशन ऑफ प्राइमरी एंडोस्पर्म सेल. दिस विल डेवलप इनटू एंडोस्पर्म एंड दिस फ्यूजन इज नोन एज ट्रिपल फ्यूजन. व्हाई दिस इज ट्रिपल फ्यूजन? क्योंकि जो मेल गमीट है दैट इज हैप्लॉइड, पोलर न्यूक्लियाई, टू न्यूक्लियाई आर देयर इन द सेंट्रल सेल. These two nuclei, when fused with the haploid nucleus of the male gamete, it will form a triploid structure known as endosperm, and this fusion is known as triple fusion. Now as this will take place, the antipodal cells will degenerate, the synergids will degenerate. Since two type of fusions are taking place inside the embryo sac, one is fusion of male gamete with the egg cell guided by filiform apparatus of the synergid. Second fusion is fusion of second male gamete with the polar nuclei. Therefore, such fertilization is known as double fertilization in flowering plants. Now here the fertilization event is shown to you. Image is depicting fertilization. Here you can see this is the female gametophyte, polar nuclei, synergids, egg cell and micropylar end. This pollen tube is guided through the micropylar end. Filiform apparatus guided through the synergids to the egg cell. One of the male gamete will fuse with the egg cell. Other male gamete will guide it to the polar nuclei. And this will result in formation of triploid nucleus. Whereas when egg cell will fuse with the male gamete, there will be formation of diploid zygote. Now fertilization, after fertilization, the events which will take place are known as post fertilization events. आपने बेटे ये चीज़ 10th में भी पढ़ी थी. तो 10th में आपने पढ़ा था कि जो फर्टिलाइजेशन हो जाती है, there is formation of zygote which will divide to form embryo, and this embryo will further divide to form seed. The ovule, fertilized ovule, will get converted into seed, and the ripened ovary will get converted into the fruit. Petals, stamens, sepals, styles, stigma, they will shrivel or fall off. बट इन क्लास 12th इनकी थोड़ी सी हम डिटेल पढ़ेंगे, एंड दैट इज द पोस्ट फर्टिलाइजेशन इवेंट्स इन फ्लावरिंग प्लांट. इन क्लास 12th आप इन इवेंट्स में उनको क्लासीफाई करेंगे. दैट इज डेवलपमेंट ऑफ एंडोस्पर्म फ्रॉम द प्राइमरी एंडोस्पर्म सेल, then development of embryo from zygote, development of seed from ovule, and development of fruit from ovary. How these developments or how these structures are formed, that will include post fertilization events. Now first step in the post fertilization event is endosperm formation. Primary endosperm formed as a result of triple fusion that will undergo repeated divisions, and a triploid endosperm tissue is formed. The main function of endosperm is to provide nutrition to the developing embryo. Here in the diagram you can see two male gametes are there in the pollen tube. One male gamete, here this is female gametophyte, egg cell and central cell. Male gametes are guided to the egg cell and central cell. Here male nuclei, one is towards the egg cell, other one is near the central cell. When these two fusion will take place, zygote will form, which will convert into embryo. Therefore it is written embryo precursor. And when the male gamete, the second male gamete fuse with the central cell, it will form the endosperm precursor. Now endosperm can be the free nuclear endosperm or the cellular endosperm. Free nuclear endosperm is the most common type of endosperm development. Here you can learn it or understand it with the help of the example of coconut. Coconut में बेटे टेंडर कोकोनट में जो पानी होता है उसके अंदर, that is the free endosperm, free nuclear endosperm. बहुत सारे न्यूक्लियाई फ्लोट करते हैं in the highly nutritious clear fluid, and when the cell walls will develop enclosing the several nuclei, these cells and nuclei start settling at the periphery and form the layers of cellular endosperm, जिसे आप कर्नल कहते हैं. कोकोनट जो आप खाते हैं, that is the cellular endosperm. Therefore in completely mature coconut, पानी जो है that will turn in milky, surrounded by cellular part, kernel, and it does not contain free nuclei. Here in this diagram, cellular and free nuclear endosperm is shown with the help of a picture or image. A image is showing the nuclear endosperm, many nuclei can be seen here, and this is then replaced by the cellular endosperm by the formation of cell walls around the nuclei. Now next is embryo. How zygote convert into embryo? The process by which zygote convert into embryo is known as embryogenesis. A diploid zygote is formed by the fusion of haploid male gamete with the haploid female gamete. This diploid zygote will divide to form embryo after the endosperm formation has started, because this developing zygote will take nutrition from the endosperm. The stages of embryo development are same in monocote and dicote embryo, and embryogeny refers to the stages of the embryo development, and it include many stages like 2-cell stage will be there, 8-cell stage, globular shape stage, heart shape stage, torpedo stage, and finally the mature embryo will be formed. Now these stages are here, you can see in this diagram. Zygote firstly will form, undergo divisions. This is the 2-cell stage. Then 8-cell stage will be formed. Then globular, heart shape or torpedo stage will be formed, which will ultimately result in the formation of the mature embryo. Now embryogenesis, how this zygote will convert into embryo. The steps are described over here. Embryogenesis, the zygote undergo asymmetric transfer cell division, जिसके कारण से बेटे 2 सेल बनेंगे. The small apical cell which is above the large basal cell. नीचे वाला सेल जो है that will be the large, known as basal cell. Upper cell is the small and known as apical cell. This apical cell give rise to hypocotyle, shoot apical meristem, and cotyledons. Large basal cell on the bottom consists of large vacuole and it will give rise to hypophysis and the suspensor. Now after 2 rounds of the longitudinal divisions, पहले ट्रांसफर डिवीजन हुई थी, अब longitudinal divisions हैं. After 2 rounds of the longitudinal division, 1 round of transfer division, an 8 cell embryo is formed and it has 4 distinct domains. The first 2 domains that will contribute to the embryo proper. The third domain, the basal embryo domain, contain hypophysis. The hypophysis will later give rise to the reticle and the root cap. And the last domain, the suspensor, it is the region at the very bottom which connects the embryo to endosperm for nutritional purposes. Now, after 2 cell and 8 cell stage, there will be the globular stage. Here the ground stem will go on to form the ground tissue which include pith and cortex. आपने +1 में अगर याद हो pith और cortex structures की हुई हैं, how it is built up in the different parts of the plant. The procambium which will eventually form the vascular tissue or the conducting tissue xylem and phloem. Then the heart stage will be formed. This is the transition period where the cotyledons finally start to form and these will elongate. And then a pro-embryo stage which is defined by continuous growth of the cotyledons and axis elongation will take place. Now, in the torpedo stage of the development, parts of the suspensor complex must be terminated. The suspensor will be shortened because at this point in the development, most of the nutrition from the endosperm has been utilized and there must be space for the mature embryo. Therefore, the maturation stage will start in which the cell growth will take place. The storage of macromolecules such as oil, starches and proteins which are required as food and energy supply during germination will take place and these result in seedling growth. Now this image depicts the 6 movements in embryogenesis and the parts involved. Here you can see the numbering Roman numerals as well as 1, 2, 3, 4. 1 is representing endosperm. Here you can see 1. This is endosperm. 2 is zygote. 3 is embryo. 4 is suspensor. Here you can see the region, suspensor region. Then 5 cotyledons. 6 shoot apical meristem and 7 root apical meristem. Now these Roman numerals 1, 2, 3, 2, 4, 5 and 6, these are indicating, 1 is indicating 2 cell stage, 2 is the 8 cell stage, 3 is the globular stage. Here you can see embryo is heart shape, 4th stage में embryo बेटे heart shape दिख रहे, तो this is the heart stage, heart shape stage, and 5th is the torpedo stage, 6th one is the maturation stage. Now the dicotyledonous embryo and monocotyledonous embryo, इनमें क्या difference है ये करेंगे अभी हम. Dicotyledonous embryo parts, it has following parts like embryonal axis, the main axis of the embryo, which will divide it into different regions like cotyledons will be there. Cotyledons are known as seed leaves which help in food storage and these provide nourishment to the developing radicle and plumule. Then there is epicotyle region which is the part of embryonal axis above the cotyledons. It terminates at plumule. Then there is hypocotyle region. You can see in this diagram hypocotyle region. This is the part of embryonal axis below the cotyledons and this will terminate at radicals. Plumule will represent the shoot tip or the shoot apex. Radicle will represent the root tip and root cap is the covering on the root tip. Now next is monocotyledonous embryo. It has following parts. One is embryonal axis, the main axis of the embryo, which it divided into different regions like cotyledons. Monocotyledonous embryo have only one seed leaf there, and that is termed as scutellum. Here you can see scutellum. It is located at one side of the axis. Then plumule region, which is representing the shoot apex. Then radical region, which is representing the root tip. Coleorhiza, which is the sheath on the radical and root cap. Coleoptile, which is the sheath enclosing the plumule, and leaf primordia. That's all for today. Let's recapitulate that what we have done today. We have studied about pollen-pistil interaction. It depends on the compatibility of the pistil for the pollen. If the pistil is compatible for receiving the pollen tube will be formed, pollen grain will germinate, tube will be formed, and generative cell will divide to form two male gametes, and tube nucleus will guide the male gametes toward the filiform apparatus of the synergids, and these in turn will guide it towards the egg cell. Then fertilization will take place. One of the male gamete will fuse with the egg cell, other male gamete will fuse with the polar nuclei. The polar nuclei, when fused with the male gamete, it will result in formation of triploid endosperm, and one male gamete which will fuse with the egg cell, it will form diploid zygote. Then after fertilization, post-fertilization events will take place, which involve the division of zygote to form 2-cell stage, 8-cell stage, globular stage, heart-shaped stage, torpedo stage, and ultimately the maturation stage, which then result in formation of dicotyledonous embryo and the monocotyledonous embryo. The detailed structures we have discussed. In dicotyledonous embryo, there will be 2 cotyledons, whereas the monocotyledonous embryo having one cotyledon which lie towards one side and known as scutellum. That's all for today. I hope you enjoyed this. Thank you.